Assalamualaikum warahmatullahi wabarakatuh. My name is Nurul Nazirah Video Osman. So from my video assignment, I choose microbial genetics as my topic. So let's go deeper about this topic. Let's see the introduction first. So what is genetic? Genetic is the study of transfer of information between biological entities, and it is very important for variation in living organisms. So where is the information? So the information located in the DNA in the nucleus, and the DNA in the nucleus will carry the genetic information and store the genetic information in human or living things. So how is the structure of DNA? So DNA is made of helical sugar phosphate backbone of two strands held together by H plus bond, and also made up of nucleotide, 5 carbon pentose sugar, a nitrogenous base, and a phosphate group. So let's see the flow of the information. So for the DNA will store and carry the genetic information. So the genetic information from the DNA will undergo transcription process and translation process to form a new DNA strand. First process, which is transcription process. So what is transcription? Transcription is the process of transfer of the information from the nucleus to the cytoplasm. To make it simple and easy to understand, basically the translation process is the process by which the information in a strand of DNA is copied into a new molecule of messenger, which is mRNA. So there are 3 steps in this process, which is initiation step, elongation step, and termination step. So the first one is initiation step is a DNA molecule is unwind and separate to form a small open complex. RNA polymers bind to the promoter of the template strand. So for elongation step is the RNA polymers move along the template strand and synthesizing an RNA molecule. So the last one is termination, which is termination is the ending of transcription and occur when RNA polymerase cross a stop termination sequence in the gene. The mRNA strand is complete and it is detached from the DNA. So basically that's all for the transcription process. So what is translation? So to make it simple to understand, we know that in transcription process, we make mRNA, right? So in this translation process, we just want to decode mRNA into a polypeptide chain of amino acid. That's all from translation. It's that we translate the sequence into a polypeptide chain. There are also 3 steps in this translation process, which is initiation, elongation, and termination. So for the initiation step, the ribosome get together with the mRNA and the first tRNA, so translation can begin. And the second one is elongation step, which is amino acid are brought to the ribosome by tRNA and linked together to form a chain. And the last one is the termination, which is the finished polypeptide is released to go and do its job in the cell. Next, we see the different type of RNA, which is there are 3 types. So the first one is mRNA. So the function of mRNA is the carry coded information. And the second one is rRNA, which is the function is the structure in which protein synthesis take place. And the third one is the tRNA. So the function is to bring specific amino acid to the ribosome, which are then meshed up to take mRNA blueprint. So just before this, I explained about the overview of genetic and how the process of the genetic information are being transcribed. So now I want to focus on bacterial genetic. So what is bacterial genetic? Bacterial genetic does not resemble the complex structure like eukaryote, and replication of bacterial DNA begin at one point and move in broad direction from there. So there are 2 types of the changes in genetic code, which is recombination and mutation. So for the first one, recombination is actually a normal condition and because of sexual reproduction. So usually in eukaryote, the recombination happen because of the sexual reproduction, and if it prokaryote, it happen through the transformation, transduction, and conjugation. So for the mutation is abnormal condition, and maybe because the exposure to agent that can disrupt the genetic code. So now we will focusing on prokaryotic genetic exchange, which is the first one is transformation. So the transformation is a bacteria, they take up DNA from their environment and incorporate it into their genome. And the transduction is a process which is the movement of DNA between bacteria by viruses. And the third one is conjugation, which is the bacterial cells, the direct transfer of DNA by bacteria, usually via plasma. So the first one is transformation. Transformation in bacteria is a gene transferred from one bacterium to another. So it's as a turn of a gene by a bacteria from surrounding environment. So in genetic cause, I learned that transformation process is like when someone die, they will leave their property like house or money. So when they die, someone will take it. So I apply for this also in this transformation when one bacteria die and our life, and the another bacteria will take up their gene. So it requires no special appendage and only involve free donor DNA fragment. So donor and recipient are not in contact with each other. So the next one is transduction in bacteria. So basically, transduction in bacteria will use a bacteriophage and virus as their vector. So once a phage infect and enter the bacteria cell, so they will synthesize their viral genome and cause the bacterial cell to lyse and release their particle that containing the bacterial DNA. So we replicate more DNA with the virus content. So the last one is conjugation in bacteria. So conjugation is made of sexual mating, and usually it occurs in gram-negative bacteria. So genetic material transfer from one bacteria to another bacteria via a direct connection. So only one strand of DNA is transferred, and the recipient complete the structure of the DNA by synthesizing the DNA acquired from the donor. So it will form a bridge between cell to transfer DNA. So the next one is a mutation. Mutation is the change in genetic material and base sequence of DNA. Gene may become less active or inactivate. So there are several types of mutation, which is the first one is base substitution. Base substitution is the replacement of a single nucleotide in a nucleic acid. And then base deletion. A deletion changes the number of DNA base by removing a piece of DNA. And then base insertion and insertion change the number of DNA base in a gene by adding a piece of DNA. In frame shift mutation, one of few nucleotide pair are detached or insert in the DNA. So the missense mutation, a change in codon that lead to placement of a different amino acid, and the last one in nonsense mutation is change in normal codon into a stop codon. So there are two types of cause of mutation, which is spontaneous mutation and induced mutation. Spontaneous mutation is a condition which a random change in DNA because of error in replication due to unknown causes. And induced mutation is exposure to known mutagen. For example, physical mutagen, agent which causes mutation by damaging the DNA. So example is like UV light or X-rays. So the chemical mutagen is to alter the chemical or physical structure of DNA. So the example, nitrous acid and acridine dye. So this is the list of reference that I used to finish this assignment. So I think that's all from me. Thank you very much to my lecturer because teaching me for this one semester, and I hope I will see you soon. Thank you very much.